Androgen is important in masculine sexual differentiation. Androgen insensitivity syndrome AIS is the result of an androgen receptor gene that is located on an X chromosome. A dysfunctioning androgen gene causes normal development of the testes, but they will not be responsive to androgen. When 5-alpha reductase is disabled, testosterone is no longer converted to NHT in an XY individual. Normal testes and reproductive structures are underdeveloped masculinized genitalia. During puberty, they become fully masculinized, and guevadoses are regarded as female when young. After developing masculinized genitalia during puberty, they are regarded as male. Behavior is more difficult to label as masculine or feminine. Almost all individuals behave in some manner typical of the opposite sex. Sex, gender, and sexual orientation are independent of one another, as shown through the various syndromes described previously. Experiments showed that females displayed lordosis when injected by ovarian steroids, but not when exposed to testosterone in utero. It shows how the brain is target tissue, masculinized or not, masculinized by androgens, and this lasting effect is called organizational effect. Steroids can have a permanent organizational effect only during a sensitive period like early development. Androgen given after birth or the neonatal period can affect behavior later in life. Full masculine behavior requires androgen during development and adulthood. Estradiol is a metabolite of testosterone. Aromatase converts testosterone into estradiol in a reaction called aromatization. Aromatization hypothesis states that during rodent development, testicular androgens are converted to estrogens within the brain, which then masculinize neurons. A blood protein called alpha-fetoprotein binds estrogen and hinders entrance to the brain. Masculinization of the brain determines male copulatory behavior, not the masculinization of the genitalia. Sexual dimorphism is a term coined by Darwin describing the conditions of males and females showing distinguishable differences in appearance, such as number of neurons, sizes of brains, and shapes of brains. Brain nuclei are 5 to 6 times larger in male songbirds than female songbirds. The muscular singing organ called the syrinx is controlled by the robustus archstriatum, RA, which regulates motor neurons in this organ. RA is controlled by the higher vocal center, HVC. Lesions of these two regions result in disrupted singing patterns or abilities. Lesions in the preoptic area, POA, in the hypothalamus of rats results in the disruption of olfactory cycles in female rats and reduced copulatory behavior in males. The sexually dimorphic nucleus of the POA, SDN-POA, is a region of the POA that is much larger in male rats. This supports the aromatization hypothesis.